Hello, I'm Jason Norman, Director of Systems Biology at Vedanta Biosciences, a biotech company in Cambridge, Massachusetts. I'm going to be telling you today about the discovery of our lead candidate for the prevention of C. difficile recurrence, VE303. VE303 is a live biotherapeutic product, or LBP, with a defined composition of 8 non-pathogenic, non-toxigenic strains of bacteria isolated from healthy human donors. I'm going to discuss with you today how we selected VE303 based on the ability to 1. Kill C. difficile, 2. Restore secondary bile acids and short-chain fatty acids in the gut, and 3. Promote the recovery of the microbiota. Each strain is grown from clonal cell banks under GMP conditions, enabling a pure and consistent drug product. The manufactured drug is stable, enabling flexible storage conditions and dosing. And I'm going to show you in the next couple of slides that VE303 is able to durably colonize human subjects. This is a figure describing the effects of antibiotic use on the microbiome and C. difficile. Antibiotics lead to a change in the microbiota community, resulting in altered secondary bile acids and short-chain fatty acids in the gut. And in this environment, C. difficile spores are able to propagate and cause disease. VE303, on the other hand, promotes the recovery of the microbiota community by both directly and indirectly leading to the recovery of secondary bile acids and short-chain fatty acids in the gut. This leads to an environment that is able to kill C. difficile, inhibit spore germination, and promote gut barrier integrity. We began by evaluating the microbiota of healthy FMT donors, rCDI patients pre-FMT, and the microbiota of rCDI patients who responded to FMT. On the top left of this heat map, you'll see a number of genera that are enriched in rCDI patients that are reduced in abundance with FMT treatment. On the bottom of this heat map, you'll see several genera that increase in these FMT responders, and these taxa form the basis for VE303. In this slide, I'm showing you that the VE303 strains provide benefit in an animal model and in vitro. On the top right, you see that VE303 promoted the survival of mice infected with C. difficile to levels that were similar to FMT and were significantly greater than no treatment controls. On the top left, in vitro, VE303 strains are able to directly kill C. difficile, and most notably, VE303 strain 6 encodes a ruminococcin gene that is known to inhibit C. diff. On the bottom left, I'm showing you that VE303 strains consume carbon sources that are known to be utilized by C. difficile, and therefore the VE303 strains occupy an ecological niche that overlaps with C. difficile. On the bottom right, I'm showing you that VE303 strains produce key metabolites that are associated with gut health, including acetate and butyrate. VE303 was given to healthy volunteers in a phase 1 study after a short course of vancomycin with ascending single-day doses or multiple-day doses. Or VE303 was administered over multiple days without vancomycin pretreatment, or as a control, vancomycin only was administered to healthy volunteers. The primary objective was to evaluate the safety of VE303, and no drug-related SAEs were observed at any of the doses. The secondary objectives were to evaluate the PK and PD at increasing doses with and without vancomycin administration. And over the next couple of slides, I'm going to share with you these data. In healthy volunteers given multiple days of VE303, 100% of the VE303 strains were detected in some subjects within the first week after starting VE303. These data are shown on the top left, where the strain detection remained high through 8 weeks and strains remained present throughout the 1-year follow-up period. VE303 strains were able to expand in abundance within the first 48 hours of starting treatment, reaching levels comparable to FMT, and the abundance remained greater than 1% through week 12, then began to decline. This decline is happening while the resident bacterial community is recovering from the vancomycin administration. To more formally quantify the differences in dosing, We have fit the strain dynamics over time for each subject to a logistic model and calculated the area under the curve. This enabled us to quantify the total strain colonization for each subject. Plotting the total AUC for all strains shows a clear dose response where multiple days of VE303 significantly increased the colonization of VE303 compared to single-day dosing. These analyses also demonstrated that vancomycin was required to observe VE303 strain colonization. VE303 also promoted the recovery of the microbiota. Here on the top left, I have taken the ratio of bacterial taxa high at baseline to those increased in abundance by vancomycin treatment. This post-vancomycin recovery index demonstrated that multiple-day dosing expedited the return to baseline community composition compared to single-day dosing. This was characterized by reductions of taxa that tend to cause infection and acquire antimicrobial resistance, including Klebsiella species. Functionally, VE303 also expedited the recovery of secondary bile acids that are known to inhibit C. difficile, as well as promote the recovery of beneficial short-chain fatty acids in the gut within the first 48 to 72 hours of starting VE303 treatment. As I've shown you, VE303 promotes the restoration of a healthy microbiota after antibiotic treatment by promoting the recovery of taxa that are associated with gut health, as well as functionally increasing the secondary bile acid pool and short-chain fatty acids in the gut, creating an environment that is inhibitory for C. difficile. We have recently completed the enrollment of a phase 2 study in the United States and Canada, and top-line results will be available in the coming weeks. In this study, VE303 was administered to subjects at high risk for C. difficile recurrence at 2 doses compared with placebo controls. And the primary objective was to determine the effect of VE303 on C. diff recurrence at 8 weeks and to determine the recommended dose for a phase 3 study. The secondary objectives were to characterize the VE303 strain colonization and changes in the microbiome. I'd like to thank you for your time.